going to be talking about observational data and other types of data to decide what works during a pandemic. So let's just suppose that there is a new virus that starts infecting humans. If that happened, and many people get sick and die, then what do we do? The obvious answer to these questions on how do we treat a previously unknown disease is, well, in the long term, we have to develop new drugs and vaccines. And in the short term, we had to check whether the drugs that we already have that work for other diseases also work for this new one. And that is the repurposing of existing drugs. The question is which drugs? There are hundreds or thousands of them that we could use. How do we choose? And after we choose, how do we determine which of them work for this new disease? That's really the topic of my talk today. The easier question is how to determine what works, because we can design and conduct randomized trials. We do trials for all these drugs that we have chosen, and we determine whether they work for a new disease. The harder question is how do we decide which drugs go into randomized trials? Because clock is ticking, people are dying. We need to be very careful about how we choose, because the first wave of trials will be very important to decide whether we're going to save a lot of lives and human suffering. We cannot study all possible drugs, so we need to choose. Also, maybe the most important question is who gets to choose? Who is going to make those decisions about which drugs go into randomized trials and is going to pay for those trials? So how do we decide which drugs go into trials? That is a question that we're dealing with here first. Well, for drugs that are not in use, exist, but are not being used, like molnupiravir, we have to use in vitro studies, in vivo studies in animals, in silico studies with model simulations. And if those are promising, then we can launch randomized trials, phase 1 or phase 2 trials first to evaluate whether they're toxic, because they're not really being used, even though exist. For drugs that are being used, like tocilizumab, then we can do all of the above, the in vitro, in vivo studies, and then we can jump, if it is promising, we can jump into phase 3 trials, because we know about their safety, because they're being used. In fact, for drugs that are in use, we could even do observational studies in humans before we jump into trials, and that is what happened with some of these drugs. So to determine effectiveness and safety in humans, not in vitro or in animal experiments, we can use observational studies for drugs that are widely used, and they can give very early evidence if we have everything set up, if we have electronic health records ready for research or national registries ready for research. If everything is ready so that the observational data can be used fast, observational studies can provide early evidence about effectiveness and safety, and they can help design better trials too. The problem, of course, with observational studies is that there may be confounding. That is, that if we find differences between those who take the drugs and those who don't take the drugs, maybe the differences are not because of the effect of the drug, they are because of differences in the people who take and don't take the drug. If that is confounding, then as we all know here, we have to adjust for confounding and try to do our best there. The other option is to use randomized trials, which is really the only option for drugs that are not widely used. Their main disadvantage is that they take longer, because the trial for a disease like COVID-19, well, may take at least months, sometimes years. So we have to be very selective about what goes into the trials. And the upside is that we don't have to worry about confounding if we can do large trials that are informative. So again, that is a question. How do we decide which drugs go into the randomized trials? What we typically do is we build evidence from inferences from various sources. And if the evidence looks promising, then we launch the trials, right? That is a theory. That is how we tell ourselves that we're making this decision. So I'm going to show you a few examples for the last couple of years, where we can see how these decisions were made based on what evidence. And we will have a discussion at the end about whether the system works or it needs some fine-tuning. Before I start with that, just a reminder to everybody that COVID-19 is a disease with two phases. It's almost like two diseases in one. The first phase is the viral infection. And there are some symptoms associated with a viral infection that are moderate or mild or even asymptomatic. And then when the viral replication starts to go down, then the symptoms start to disappear. And here, for this phase of the disease, there's the potential benefit of antiviral drugs that block viral replication in some way. Then, for some people, fortunately not for most people, there's a second phase of the disease when there is a hyperinflammatory response. This happens even after viral replication is declining. And this is what leads to the severe symptoms, to the acute respiratory distress, and can lead to death. So for that phase of the disease, antivirals are not the drug of choice because this phase of the disease is relatively independent of viral replication. So here is where the anti-inflammatory drugs will be most helpful. For example, I just mentioned tocilizumab or dexamethasone. Because we only have a few minutes to talk about this, I'm going to focus on the antivirals, not on the anti-inflammatory drugs here. And I'm going to talk about 4 of those drugs. The first one is, well, first I'm going to talk about drugs that existed in 2020, but they were used by few people at that time, which means that we cannot really do observational studies with them. One drug was remdesivir, which has a safety profile, not very well known because not many people have used it in the past. We'll talk about that more. And hydroxychloroquine, which has a very well-known safety profile because even though it's not very much used now, it has been used for decades to treat malaria. Then I'm going to talk about one drug that existed in 2020, but had really never been used, molnupiravir. So that drug, we didn't know the safety profile very well. And finally, I'm going to talk about one drug that existed in 2020 and was being used by many people, tenofovir. And that's a drug with a very well-known safety profile and for which we can actually do observational studies. So let's start talking about remdesivir. This is a drug that was developed by Gilead and was studied as a treatment for Ebola virus, among other things. This is a drug that interferes after being metabolized. It interferes with the viral RNA-dependent RNA polymerase. So there is a blockage of the chain, and the enzyme cannot keep making RNA, so the virus cannot replicate. And is active against a broad spectrum of viruses. And the only problem is that it can only be given intravenously. There is not an oral formulation. This was one of the first drugs that was approved for use in humans. In May 1, 2020, the FDA issued an emergency use authorization for remdesivir in hospitalized patients with COVID-19. So the first drug for which we must have had some evidence that it worked because the FDA was doing it. It was very expensive, more than $3,000 per treatment course. So there were a few key randomized trials of remdesivir in hospitalized patients. They were all done before the Delta variant of SARS-CoV-2. The first trial, which was finished by May of 2020, which is a trial that was used by the FDA to make the decision that they made. This trial, with about 1,000 people, found a shorter recovery time and have a mortality relative risk of 0.7. Then there were at least 3 other trials, bigger trials. The first one was the Solidarity trial. I talk more about that. That is a trial that was conducted by WHO, and that trial didn't find any effect on mortality for remdesivir. That was finished in October 2020. Then there was another trial, the Discovery trial, that also found not much of an effect on mortality. And the final trial that has been published very recently found a relative risk of 0.8. So these findings are compatible with a modest effect on mortality for hospitalized patients, not very big, but if you don't have anything else, I would want to be given remdesivir. So why did we have so many randomized trials of remdesivir? Well, because WHO, the World Health Organization, said that in March 2020. In February of 2020, there was a program that WHO started to make recommendations about drugs that should go into trials quickly. And in March 2020, the scientific advisory group made the recommendation of studying 2 drugs, remdesivir and lopinavir. I've been trying to find out who the people in scientific advisory group were, but I cannot find it. That's what the picture says on the WHO website about the names of those people. So I'm sure that they were great experts, but I cannot credit them because they're not named in the report. But they made this recommendation. And in March, the WHO Solidarity trial was launched with 4 active treatment arms. They had 1 arm for remdesivir, 1 arm from lopinavir, 1 arm from hydroxychloroquine, and 1 arm for interferon, plus controls. The arms of hydroxychloroquine and lopinavir were stopped in the fall because they didn't work. And only these 2 arms were continued. And as you saw, they didn't find that they worked either at the end of the trial. But why? Why was remdesivir considered for immediate evaluation by the WHO? So this is what the Scientific Advisory Committee said. They said that it was a first priority. That was the first drug that we have to look at because of the broad antiviral spectrum, because of the in vitro and in vivo data. And there were some studies that shown in vitro activity against SARS-CoV-2 that was published in February of 2020 by Wuhan investigators. And also, there was also some information at that time about in vivo activity in animal models against other coronaviruses like MERS and SARS-CoV-1. And also because of the extensive clinical safety database, which was based on patients that have been treated for Ebola in the past. I don't know if I would say it was very extensive when you compare with other drugs that are more in use, but there was safety data and it didn't seem to poison people. So think about this now with the benefit of hindsight. we are 2 years later, and you see how humbling this exercise is. I mean, you are in a scientific advisory committee. You have to make a recommendation of which drugs to use. It wasn't easy based on the data that you have at the time. So in fact, after that decisions and the Solidarity trial is launched, of the 4 drugs that they tried, 3 didn't work, and 1 had a modest effect. So it's a very hard job. I don't envy the people who had to make those decisions. And the problems that they had was, one, that there was not much data relevant to SARS-CoV-2. So you have to use some in vitro studies that people have done quickly before March 2020. I mean, the virus was identified in January. So that was not much time. And also the trials were being done in hospitalized patients. But see, we just said that the disease has 2 parts, and it's not clear that antivirals work in established disease after people are at the hospital. Maybe they should be given earlier. That's what happens for other diseases, right? In fact, that is what happens for Ebola, for influenza, and for HIV. You start treating early, you have better clinical outcomes. And in fact, there was a study that was published in April of 2020 of a study of remdesivir in monkeys, and it showed that had a clinical benefit. But the monkeys were given the drug 12 hours after inoculation of the virus, not 1 week later. So a randomized trial was done for non-hospitalized humans. And that trial, which ended in the spring of last year, almost a year ago, was a trial in high-risk individuals, people at high risk of bad COVID outcomes because they had risk factors. And that trial, a mortality hazard ratio of 0.13. So bingo, finally, we find where it works. It works for people who are not at the hospital shortly after developing symptoms. So the WHO committee wasn't that misguided. They were using the data that they have at the time, and they were making a recommendation. The problem was that we were doing the studies in people who were not likely to benefit much because they were in the inflammatory phase of the disease, not in the viral replication phase. And that is still a problem. This is an expensive thing, and it's intravenous. So we have to give this treatment to people who have very few symptoms. We have to convince people who have COVID-19, not many symptoms, say, well, if we give you this, you have a much lower risk of death, but your risk of death to start with is very low. So it's not an immediate, it's not an easy decision whether you treat a patient or whether you accept to be treated. Okay, I told you that hydroxychloroquine didn't work. Let's talk a little bit about hydroxychloroquine. Because, yeah, there are multiple randomized trials for the treatment of hospitalized patients. It doesn't work. I mean, case closed. We don't need more trials of this. It doesn't work. But again, these are hospitalized patients. And what if hydroxychloroquine works for early treatment? No, we just saw that that is what happens for remdesivir, right? So we could look at the trials that were conducted for prophylaxis of COVID-19 using hydroxychloroquine. There are 2 types of trials of prophylaxis. Those that are pre-exposure, so you give the drug to people before they are exposed to the virus, and the post-exposure, which is kind of early treatment, right? You give it after they have been exposed and they may be infected. So there are several trials that look at that. So we did a meta-analysis of these trials. And this is what we found. There are 7 randomized trials of hydroxychloroquine for pre-exposure prophylaxis of COVID-19. The pooled risk ratio is 0.72 for COVID-19 diagnosis of the trials. And the confidence interval, you cannot see it well, goes from 0.58 to 0.9. Some people say that we were not using the right random effects estimator, that we should method, that we should use a different one. We also use it, and the confidence interval goes from 0.55 to 0.95. So hydroxychloroquine, based on the randomized trial evidence, works for the prevention of the pre-exposure prophylaxis of COVID-19. I mean, we thought that it didn't work. What happened here? Right? Well, several things happened. First, it doesn't work for the treatment, for clear, and that kind of color the way we were looking at these studies. Also, the first studies were post-exposure prophylaxis, not pre-exposure. So maybe the drug needs some time to reach the blood levels or whatever levels it needs to work. But the most important problem that happens with hydroxychloroquine is that the trials were small. So they found results with very wide confidence intervals. And they were published in the best journals, in the New England Journal of Medicine in the summer of 2020. The hazard ratio was 0.7, but the confidence interval was very wide. So the author said, hydroxychloroquine doesn't work. And the editor said, see, this proves that it doesn't work. And the environment and the fact that some prominent politicians were talking about it didn't help in trial after trial, because each trial was very small. We concluded that it didn't work. Let me give you one extreme example. That was a trial published in September 2020 in JAMA Internal Medicine. This trial found no significant difference for hydroxychloroquine compared with placebo. But there were 64 people in each arm. 64 people. There were four cases in each arm. They didn't even give the risk ratio. I'm adding it there for you based on the data from the trial. The risk ratio was 0.95. Look at the confidence interval. From 0.24 to 3.64. If you had to find the definition of a non-informative randomized trial, that's it, right? You cannot say anything about that trial. All you can say is that the findings from this trial are very compatible with an effect that goes from 0.24 to 3.6. So that's exactly the same as saying 0. I know nothing about this. But that is not how this trial and all the other trials were presented in the media. Let me give you an example. This trial was done by people at the University of Pennsylvania. This is what the press release from Penn said. Hydroxychloroquine, no more effective than placebo in preventing COVID-19. The trial was completely non-informative, and the results were being presented as no effect. Not statistically significant doesn't mean no effect. It means you don't have enough data. So wait until you have more data. So hydroxychloroquine daily did not reduce the risk of infection, really. In fact, if you do a cumulative meta-analysis of the randomized trials of both pre and post-exposure prophylaxis for hydroxychloroquine, which we did, and I'm showing you here, you see that in the fall of 2020, there was already enough evidence to show a protective effect of hydroxychloroquine for the prevention of COVID-19. And that became, the confidence interval got narrower and narrower over time. So why is this a problem? Well, this is a problem first because we convince ourselves that hydroxychloroquine doesn't work for the prevention of COVID-19. And you could say, well, yeah, but I mean, shortly after the fall of 2020, we had vaccines was better to have vaccines that having a treatment with the hazard ratio of 0.7 for COVID infection. We don't know what the hazard ratio is for mortality. Maybe it's much stronger. We don't know. But the problem, what happened by doing this incorrect interpretation of lack of statistical significance as no effect, is that the randomized trials of hydroxychloroquine that were ongoing could not finish because we told people it doesn't work. And now the investigators go and try to convince individuals to participate in a trial of hydroxychloroquine. People say, no, thank you. It doesn't work. Didn't you read the news? So most of the later trials of hydroxychloroquine could not be finished, which means that they couldn't get precise estimates, which means that we could not see what was really happening until we put all of them together. So this had misinterpretation of statistical significance by authors and editors and the media had very concrete implications for how we do science. And I will explain later how this effect was even more severe for other drugs. Okay, so let's talk now about molnupiravir. So when the pandemic started, molnupiravir was in preclinical development, and it was being developed as a drug against the flu, and now it's commercialized by Merck. And this drug, after being metabolized, it inhibits the replication of the RNA viruses, but in a different way, not like remdesivir. It does it by creating a mutation. So the RNA polymerase is creating an RNA that has a mutation, and then the virus doesn't work, and then that. So it's a antiviral activity by lethal mutagenesis of the virus, and it's given orally. So how was the evidence for molnupiravir built so that we had trials? Well, first, there were some in vitro studies, and already in April 20, there were a study that showed that molnupiravir inhibits SARS-CoV-2. By the way, in January of 2022, a couple of months ago, Merck said also that there are some studies that show that it inhibits the Omicron variant. Then there was some study in ferrets, in animals, and this study also showed that molnupiravir blocks the transmission of the virus among ferrets. So with this and similar evidence, Merck decided to launch phase 2 randomized trials. 2 phase 2 randomized trials were launched in October of 2020. The first one was in hospitalized patients. It was known as move-in. No clinical benefit was found. And in April 2021, Merck decided they were not going to proceed to phase 3 with molnupiravir in hospitalized patients. Just it doesn't work, same as we saw earlier. The second study was the move out, and this was a study in non-hospitalized individuals. And in April of 21, after the promising results for the phase 2 trial, there was a decision to continue into phase 3 with about 1,400 people. So the results were first announced in October of 21 with about half of the sample, and Merck said that the risk of hospitalization or death was reduced by approximately 50%, which is a lot in people who are not hospitalized yet. As a result of that, a few weeks later, the drug was authorized in the UK. The final results were published in mid-December. And by then, the effect have gone down to 30%. Not clear why that happened, but it was not 50%, it was 30%. But like a week later, the FDA gave it an emergency use authorization, that the same that gave to remdesivir in May of 2020 for hospitalized patients, and then for non-hospitalized patients, now was giving this authorization for molnupiravir for non-hospitalized patients. Then something that may or may not be important here, but I want to talk about it, is what are the financial aspects of the approval of molnupiravir. So this drug was developed by a nonprofit at Emory University in Atlanta, and it was developed with federal money. Taxpayers' money was used to develop the drug in different grants. About $29 million at least were given in grants by NIH and other agencies to support the development of this drug. Then in March 23 of 2020, Emory sells the exclusive license to a for-profit company, to Ridgeback Therapeutics, because a nonprofit cannot really fund trials and do all the work to do the trials. So they gave the exclusive license to a for-profit company to do the trials. It is not known how much money they got for that. In early April of 2020, Ridgeback goes to the US government and say, hey, could you give me some money to do the trials? And the US government says no. So the following month, Ridgeback sells the drug to Merck. It's not known for how much money. And in June 2021, after the phase 2 results were published, the US government signs that they're going to pay $1.2 billion to Merck if molnupiravir receives FDA's emergency use authorization, which it got 6 months later. That's a contract for 1.7 million doses at $700 per dose. An independent study that looks at the production costs for molnupiravir says that it's at most $17 or $18 per course. And you can see here that the US government did a very, very bad deal. It paid for the drug, the $29 million, and then paid an exorbitant amount of money to be able to use it. As I said, this may or may not have any implications for what I'm going to discuss later. Now, why did the US government say, no, we are not giving you money to do randomized trials of molnupiravir? Because they had safety concerns. This drug is mutagenic. That means that first, it could contribute to the emergence of new variants of SARS-CoV-2 because it attacks the virus by mutating it. And two, in theory, it could be incorporated into the DNA of the host, of the human, and create a mutation, especially in rapidly developing tissues or in fetuses. And in fact, there are some animal studies showing the reproductive toxicity. So the US government said, no, we're not giving you money to do the trials until you bring us more evidence of safety. There are some structurally similar compounds to molnupiravir that were abandoned in the early 2000s because of concerns about mutations. Now, when the FDA gave the approval, the temporary approval in December, it said that the law of genotoxicity, the risk of genotoxicity is low because the treatment course is only 5 days. So there's not really much time to create that. We don't know, but it makes sense that it's only 5 days. But it asked Merck to establish a system to monitor the emergence of variants, and also did not authorize the drug for children. It's more, when NIH had to make a recommendation for the use of molnupiravir, which was updated on January 19th, actually of 2022. They said that women should abstain from sex, or they should use contraception for up to 4 days after receiving molnupiravir, and men for 3 months. So you take the drug, you're gonna have sex with a fertile woman, or you have to use some type of contraception for 3 months after receiving that. So there are some concerns. I think it's safe, it's fair to say that people are concerned about the safety of this drug. So we have this drug that is oral, which is good, moderately effective, and expensive, with some safety concerns. Okay, let's talk about our last example. Tenofovir. Tenofovir is a drug that is used as treatment for HIV, is both for treatment and prevention of pre-exposure prophylaxis of HIV, and it's also used for the treatment of chronic hepatitis B infection. Has been used for more than 20 years, is oral, is very safe, even during pregnancy, you can take it. And it's only if you are taking for very, very long periods of time in a very small percentage of patients, they can have some renal disorder that quickly reverts after stopping the drug. It also interferes with the viral RNA polymerase. And there are 2 forms of tenofovir, both have been developed by Gilead too. The first one is when the drug is given as tenofovir disoproxil, TDF. That is the form of the drug that has been used for the longest, and in fact now is off patent. It's very cheap. Any company can make it if they want them. And this drug is more bioavailable in most tissues. Then Gilead did a different form of tenofovir, is tenofovir alafenamide, which is TAF, TAF, and that's on patent yet, and is the most commonly used form now for HIV patients in many countries. They are moving from TDF to TAF because it concentrates in lymphoid tissues, and that's in principle better because there is a lower risk of toxicity, although there is a very low risk of toxicity anyway. Now for COVID, that's not very good. We don't want a drug that goes only to lymphoid tissues. But why would we consider tenofovir in the first place as a drug for repurposing? Well, it was already considered for a treatment of SARS-CoV-1 when that pandemic didn't happen, and it wasn't used because, fortunately, it didn't happen. But also, our colleagues, our clinical colleagues in hospitals in Spain, they told us, look, you know, my patients with HIV are not getting serious COVID. They noticed that in March. So it's not, I mean, they are immunosuppressed, right? So you might expect that they will get more serious COVID, but they're not. So that was a clinical observation that piqued our interest. So let me tell you a little bit about the evidence for tenofovir and COVID-19. There were some in vitro studies, but the results were mixed. Some of the studies found that tenofovir had an effect on SARS-CoV-2 in vitro. It depended on the experimental conditions. And there is at least one study that didn't find it. It's a study that has not been published, as far as I know, was an abstract in a congress in October of 2021. It's been widely cited, which is why I'm saying it here. All the authors are Gilead employees, and that didn't found any effect, any in vitro effect. There is one in vivo study in ferrets too, that in ferrets that were treated with different antivirals, including TDF, which, by the way, TDF is always given with another drug, which is FTC for treatment of HIV. Those are given together. So in ferrets they were given either TDF, FTC, or lopinavir, or hydroxychloroquine, and they found that only the TDF group had a reduction in clinical symptoms. So they said when the authors published this in the first half of 2020, they said that TDF may be the most likely candidate for SARS-CoV-2 infected host. The difference between tenofovir and the other drugs is that we have human studies, because this is used by millions of people. So we can do observational studies of TDF and COVID-19, both for people who are being treated with it and for people who are using it as pre-exposure prophylaxis. Not the first one, but one of the first ones of these studies was the Spanish HIV COVID-19 collaboration, which was an observational study of about 77,000 people with HIV in Spain in around 60 hospitals. And this study, which was published in the summer of 2020, found that people with HIV who were taking TDF FTC had the lowest risk of hospitalization for COVID-19 that people with HIV that were taking other drugs. So that was interesting, but it's hard to interpret because, well, maybe people who take TDF FTC are different than people who take the other antiretrovirals, and maybe they are healthier people and they're giving them TDF because they are healthier and they're giving the other antiretrovirals to the people who are sicker. And that would explain that. I mean, that is possible, and that study could not adjust for comorbidity or for clinical status. It didn't have any data on that. So sure, that is the possibility. What the authors did, what we did was we did a subset analysis in people younger than 60 years, because those people don't have a lot of comorbidities on average. So if the effect is still there, I mean, if this hazard ratio of 0.5 is there, it weakens the case against confounding by comorbidities. And yes, it was there. But this is not a very good proof. This is sort of just some, it might. Something that we did too is that what happened is that some hospitals in Spain, some hospitals tend to use TDF, and other hospitals tend to use staph, which is the other form of tenofovir, for which we couldn't find any inverse association with COVID risk or hospitalization. So that the hospital uses one or the other has nothing to do with the clinical status of the patients. Is that some hospitals have decided to use the more expensive drug for whatever reason, and other hospitals have decided to stay with the cheaper drug. So, but the type of patients that go to all the hospitals are similar. So we compare hospitals that have that use mostly TDF with hospitals that use mostly TAF, and the rate ratio was still 0.8. But now we have to remove a lot of hospitals. We're using both. We have a lot of misclassification. So that is not confounding, but that is hard to interpret. This wasn't a definite study. At the same time, other studies have been done in other places. So there was a study in South Africa that found that the mortality hazard ratio for TDF versus other antiretrovirals were 0.42. So people who were on TDF have less than half the risk of dying from COVID than people who were on others. And this study could adjust for comorbidities. There was another small study among users of TDF for pre-exposure prophylaxis in Madrid. It also found that the risk of COVID symptoms was smaller, was lower for people on TDF versus staff. So this is kind of nice, right? Because we have the in vitro studies that are not completely, they're not definite. We have the in vivo studies with ferrets. We have the human studies, the observational studies that are not perfect, but it's a possibility of confounding. And also TDF is cheap and has an excellent safety profile, can be used even during pregnancy, as I said. So it seems that the next step is to do randomized trials. I mean, I would say that this is a strong case. The data that I've shown you, the evidence that has been, it makes a strong case for randomized trials of TDF. I don't think it's much weaker than for other drugs that have been studied in trials. So let's look at the trials. Unfortunately, there are not many. There was one trial in hospitalized patients. The trial is not yet published. The results were reported at the CROI meeting last month, and there was no clinical benefit. So same as for all the other drugs, no clinical benefit for any antiviral so far, except maybe for remdesivir in hospitalized patients. Then there was a very small trial, like of 60 people, 30 in each group, and in France that was supposed to be just a pilot project, and that study found that in non-hospitalized patients who got TDF, they had a faster clearance of the virus in the nose. But doesn't say much. It's not. I mean, it's only 30 people with no clinical outcomes. And then there was a third trial that we did in Spain and Latin America for pre-exposure prophylaxis. Its study was designed in April of 2020 with the little data that existed at the time. So this study has 4 treatment arms. Again, this was designed in April 2020. So the arms of the trial are TDF and hydroxychloroquine, which was in March of 2020, if you can go back there, if you were going to do a trial and it didn't include hydroxychloroquine, it was like, why you are not studying that? It's like the drug. Of course, it didn't work later, but we didn't know. So this trial has 4 arms, TDF only, hydroxychloroquine only, both, and none of those. And this was done in healthcare personnel in several countries. It started in April in Spain, in October in Bolivia, and in March in Venezuela. And this figure shows you the recruitment. The recruitment for the trial in Spain in April, that's the pink columns, it was great when it started, and then it died because of hydroxychloroquine. Who wants to be in a trial of hydroxychloroquine, right? Our colleagues in Latin America did a little bit better, but it was very hard to convince people to be part of this trial. And then the vaccines came and healthcare workers were being vaccinated. So the trial was supposed to have 4,000 people, 1,000 in each arm. It ended up with 900 people, which is a very large trial, but not enough for the outcome that we were looking at. For pre-exposure prophylaxis trials, you need a lot of people. So we ended up with about 200 people in each arm. And these are the results. It's very non-informative because we ended up with 3 cases of COVID-19 in each of the active treatment arm, only 3 cases. And if you look at the risk ratio at the end there, yes, the risk ratio is 0.4, but look at the confidence interval. It goes from, essentially from 0 to 2. So not very informative trial, very disappointing. A lot of work went into this, but because hydroxychloroquine trials were interpreted as it doesn't work, we were screwed. So we had the problem there. If we combine the groups with TDF and compare with the groups without TDF, the risk ratio is 0.5. The confidence interval is a little bit tighter, but it's still very non-informative. We're at 0.5 now. So it was a great trial, I promise, but we didn't get all the people that we needed here. So this was compatible with the protective effect of both TDF and hydroxychloroquine, but the estimates are very imprecise. On the good side, on the right side, it was very. We showed that both treatments were very safe as expected, not more adverse events than the placebo arm. But look at this. Those who have heard me before know that I make a big deal of not using the concept of statistical significance in science, because it's not a scientific concept. It's something that was very unfortunate, that was introduced in science in the 20th century, and I hope that 20 years from now, no paper has the term this is statistically significant and this is not statistically significant, because it's not helpful. And not only helpful, in this case, it led to a lot of trials not being able to finish because of the incorrect interpretation of non-statistical significance has no effect. So what else? We did a couple of observational studies more, because of course, in the one that I showed you from Spain, we could not adjust for comorbidities. So we decided to do another observational study in which we would collect detailed clinical data on each of the patients. That study is known as COVID. And this study in 69 hospitals in Spain was funded by NIH and ended up with about 51,000 people with HIV. We had detailed clinical information on their baseline characteristics. The TDF group had slightly lower prevalence of hypertension and diabetes and chronic renal disease as expected, because people on the TDF group were a little bit healthier. So we could adjust for all of that. And we found that, I'm going to jump here, we found a lower risk of hospitalization from COVID-19 on the TDF group compared with, for example, the TAF group after adjustment for clinical characteristics. The risk ratio is 0.66. We found an even stronger effect for admission to the intensive care unit with the risk ratio of 0.28. And for death, there were very few deaths, so really we cannot say much. That was in Europe, so we decided to do another of these studies in the US. So working with our colleagues on the Veterans Agents Cohort study, which is a study of US veterans that are followed, and these are with HIV, mostly males here. So we compared also the risk of COVID-19 according to the antiretroviral treatment that they were using. And so we had here, we restricted our study to males, same as in the Spanish study, people with adequate virological control, so people who are not immunosuppressed. And we followed them between February of 2020 and October of 2021 without previous vaccination and all this. So we ended up with about 20,000 people with HIV. only 5% were in the TDF arm because, as I said, many healthcare systems and hospitals are moving from TDF to TAF now. We also found that people in the TDF arm had a lower percentage of renal disease, 4.5% versus 8.8% in TAF. In this study, we even have information on the GFR, so we could adjust for chronic conditions very well. We could even exclude people with chronic conditions. Look at that. And we estimated the 18-month risk of different COVID-related outcomes. And this is what we found. We found, if you look at hospitalization, again, people on TDF have a lower risk of hospitalization for COVID after adjusting for everything that you can think of. So let's review the situation. We're getting close to the end now. For these 4 drugs, for remdesivir, we have in vitro studies, in vivo studies, and some safety data. And as a result, we launched multiple randomized trials, and one of them in non-hospitalized patients by Gilead. For hydroxychloroquine, we have in vitro studies, lots of safety data, and as a result, we launched multiple randomized trials in hospitalized patients. For molnupiravir, we have in vitro studies, in vivo studies, safety concern, as a result, we have 2 randomized trials, one in hospitalized patients and one in non-hospitalized patients. Both are large and give informative answers. For TDF and FTC, we have in vitro studies, in vivo studies, an excellent safety record, and highly supportive human data from observational studies. And as a result, there are no trials in non-hospitalized patients. I hope that you share the surprise with me. Because where are all the randomized trials for TDF? And see, if you think that the observational studies are not enough, let's say that you tell me, they may be confounded. Okay, by what? What else is left for a drug that we know its prescription patterns very well, and we have adjusted for everything that in the real world doctors use to prescribe it? We have talked with dozens of experts. There's nothing else that doctors use except the things that we have adjusted for. So it is possible that the observational studies are biased for some reason that we don't know. But what is your alternative explanation for the results for the human studies? That's really the question. We need to treat this like scientists. We cannot say observational studies may be biased because of confounding. Yeah, of course. That's not a scientific statement. That's a logical statement. They may be biased. Yes, it may happen. But why? What is your theory? What's your alternative explanation? Because if you have one, we'll be happy to test it in our data and to see where it can explain. We've been doing that for the last 2 years, and we cannot find any alternative explanation. So the problem with the tenofovir story is that it encapsulates many of the misconceptions about observational data. And even in a case where we have really strong evidence that this might work, we're not doing trials. Why? So is this a missed opportunity? We may have missed the opportunity to do trials for tenofovir. It will be very hard to do them now, right? What are we going to compare them with? Nothing when we have molnupiravir and remdesivir. And just to be clear, I'm not saying that TDF works. It may or may not work for COVID-19. We don't know for sure. What we know is that the evidence to launch randomized trials of tenofovir is not weaker than the evidence to launch trials of other drugs. In fact, I would argue it's stronger because we have a lot of human data that we didn't have for the others. And think about how crazy this whole thing is. The US government is paying $2 billion for molnupiravir and was willing to pay that even before it has been proven to work in non-hospitalized patients. and is not willing to pay, what, 50 million for a randomized trial of tenofovir? This is the situation in which we are, that our system to select drugs for repurposing, even in the middle of a pandemic when the stakes are very high, doesn't seem to work perfectly. And bear in mind that we're talking about a drug that is very safe and very cheap. And I wonder whether that's part of the problem here. So thank you very much.